Okay, this is about cellular control. What is an operon? The binding site for a repressor protein, any group of genes responsible for the metabolism of lactose in prokaryotes or eukaryotes, or C, a cluster of genes under the control of a promoter, or D, a regulatory gene. So which is the correct answer? Pause the video, have a go, then unpause and watch the explanation. Okay, I'm going to go through the explanation. So on your DNA, you can have a stretch of the DNA that is controlled. And what you have are things called structural genes. So I'm going to label these X and Y, and these are known as structural genes. These structural genes have the code for the useful protein that we're talking about, okay? And they're called structural genes. Now, next to them is a gene with an O on it. And a lot of people might say, well, this is where the repressor protein binds, and that is true. So we might think that that is the operon, but it's actually not. This is known as the operator region. So this is not the operon, this is the operator, and that might screw people over. Over here is the promoter region, which is next to the operator region. Now, somewhere over here, we also have a gene for a repressor protein. So this is the gene for a repressor protein, and that is over there. So actually, let's move this over here. Okay, so how does this all work? We have something called RNA polymerase, and you probably remember that RNA polymerase is the enzyme needed for us to transcribe and make some mRNA. So to transcribe the DNA and make some mRNA. And so here's our mRNA polymerase. And RNA polymerase attaches to the promoter region. This is its binding site. However, this means that the RNA polymerase is going to read this X and Y structural gene. It's going to move down the DNA and read X and Y and produce a stretch of mRNA, a section which will be spliced and then go to a ribosome and be translated into the particular polypeptide or protein that's wanted, right? So that could be anything. It could be amylase, which is a enzyme. It could be insulin, which is a hormone. It can be any useful protein in the body that we're making. Now, how does the cell keep control of that and make sure we don't make too much? Well, this gene over here makes a repressor protein. Now, the repressor protein can bind to the operator region, and if it does that, it blocks RNA polymerase, prevents it from attaching to the promoter region. And that means that the X and Y structural genes are not transcribed and we don't get mRNA formed. So when we read this question, you'll be forgiven for thinking, well, the operon is the binding site for a repressor protein, because I've just shown the repressor protein binding to this region with the O, but this is not the operon. The O here is actually called the operator region, and so that is the incorrect answer. Any group of genes responsible for the metabolism of lactose in prokaryotes or eukaryotes. Now, the example in OCR we give you is about the lac operon, the set of genes that regulate the transcription and then translation of lactose. Okay. However, that's just an example. Many other proteins are controlled by operons. A cluster of genes, so this is obviously not correct as well because it's too specific. A cluster of genes under the control of a promoter. Well, that's actually true. The promoter region controls whether or not these structural genes get read. And so we call this whole region is known as the operon. And that is known as the operon. So the correct answer for this is actually C. And this is the one you should have gone for. And there we go. And D, a regulatory gene. Well, the regulatory gene is just over here, just before the operon. And that's what is transcribed to produce the repressor protein, which helps, well, the repressor protein, you can get activator proteins as well, or repressor proteins, which inhibit transcription, but you can get activator proteins that increase the likelihood of transcription as well. They increase the ability of RNA polymerase to bind. This particular one's a repressor, so it prevents RNA polymerase binding. All right, let's move on. Okay, number two, the table shows the genetic code in a short length of DNA, the corresponding codons on mRNA, and the anticodons on the corresponding tRNA. So what we're looking at here is a stretch of DNA. Does it match up to the mRNA? And does that mRNA, is it complementary to the tRNA? So you've got to check you've got to check this original DNA to the mRNA. Does that fly? And then does the mRNA, the codons on the mRNA, will they bind with the anticodons on the tRNA? If so, we've got the correct answer. So let's go through the rows. Now, there are quick ways to cancel some things out here. So let's check it all out. First of all, I know that RNA, remember, contains, oh my gosh, contains, whatever. RNA contains U instead of T. So let's go through and make sure you pause the video and have a go, by the way. Go through first of all and cross out any RNAs with the T in. So look, this mRNA here for A, this has got a T in it. There shouldn't be any T's in the mRNA or the tRNA. Okay, that's good. No T's, no T's. Ah, we got a T over here, GTA. That's on that's on RNA. RNA doesn't contain thymine, so these two are definitely incorrect. We don't even have to go through and match them up. So we're now dealing with just two. So there's a quick shortening of the answer. Right, let's look at B. CAT, does that go with GUA? Yep, that does, because adenine binds with T, which would be uracil on mRNA. So that's cool. AAT, go with UUA. Yep, that's correct. ACG, does that go with UGC? So A would bind with uracil, C cytosine with guanine, and G guanine with cytosine. So the original to the mRNA is correct here for B. Let's check out the tRNA connection. GUA. So G and C go together. U goes with A goes with U. That's all good. UUA, that looks all good. And ACG. ACG. Yeah, that's all correct. So our correct answer here, hopefully you've been able to see there, is B is the correct answer. Now, why? Let's spot the mistake here. We got CGC. Look, there's the mistake. Can you see this mistake on this one, which means that this is incorrect? We've got CGC on the mRNA and then CGC on the complementary bases to the CGC there. That is not correct. It should be CGC and then this should be GCG. So this is definitely incorrect. And our correct answer here is B. Let's move on. Now, if you're enjoying the walkthrough of these cell control or regulation questions, make sure you hit the like and subscribe to the channel. All right, let's move on. Boom. Eventually, I would love to move on. Right, what we got here. The bacterium E. coli produces lactase only when the sugar lactose is available. The mechanism to control production of lactase is called the lac operon. Remember, we've just been looking at an operon, so I'm not going to draw it all out again. Statements 1 to 3 describe the role of molecules in the way lac operon controls lactase production. Lactose binds to the repressor protein. The repressor protein binds to lactase. Lactase leaves the cell. Well, what we're really looking at here is, if you remember from the previous video, so you might want to pause and have a go now. If you remember from the first question, what we had is our structural genes over here, X and Y, then we had O, and then we had P. What actually happens is we have a repressor protein that binds to the operator region and prevents lactase being it prevents the lactase mRNA being transcribed and read and produced. Okay, so this can't be produced if the RNA polymerase cannot bind to the promoter region. Well, what we find is when lactose is present, the sugar, it acts as an inducer. And what it does by doing that, it binds to, similar to an allosteric site on an enzyme, it binds to part of the repressor protein. And what that does, that removes the repressor protein from blocking the region, and therefore our RNA polymerase can bind to the promoter region and then transcribe X and Y, or whatever these structural genes for lactase are. Okay, so which of these is correct? Lactose binds to the repressor protein. That's absolutely true. I just showed that. Lactose is this red triangle here. So that does occur. So 1 is definitely in our correct answers. The repressor protein binds to lactase. No, that's incorrect. We can see here, this is our repressor protein, and it doesn't bind to the lactase. The repressor protein binds to the O region, the operator region, which then blocks the promoter. Lactase leaves the cell, but we're dealing with E. coli here. The lactase is going to be within the E. coli, is produced. Once it's produced on the ribosomes, it's staying in the little single-celled organism there where it can be used. Okay, so the answer is 1 only, and that is going to mean our answer is D. So hopefully you guys are all cool with that. Make sure you hit the like. Let's go. Let's move on. Let's move on. Oh my gosh. Okay. Below are 3 processes that occur within living organisms. We've got apoptosis, we've got mitosis, meiosis. Which of these processes is important in determining the body plan of an organism? So the body plan of an organism, well, 1, 2, 3. Well, why do you not have webbed fingers, or why do you have your eyes in the right place? And apoptosis is involved a lot here, and that is cell death, controlled cell death. So our fingers start off webbed, and at some point when we're a fetus, the genes produce a protein that triggers apoptosis that kills the cells between the fingers, enabling our fingers to no longer be webbed. So apoptosis is definitely part of this. Also, whenever you're growing parts of the body, you're going to need mitosis. Mitosis is involved in the body plan of an organism, also growth, repair, and asexual reproduction. Meiosis is only involved in the formation of gametes. It's not part of the body plan, deciding where your head and your ears and your eyes go. So meiosis is not correct. So we're looking for 1 and 2 only. The correct answer is B. All right, moving on. Okay, the table below shows the stages in the life of the cell cycle. Which row A to D shows the correct order of the different stages? So what you need to appreciate, this is a cycle, so it can start with any one. You just want to make sure it's all in the correct order. So hopefully you know the cell cycle off by heart. By the way, pause the video, have a go. Okay, here's the answer. Right, I'm going to show you the cell cycle and then see if you can work it out from this. So here's our cell cycle. Of course, we need some mitosis in the cell cycle. Um, After mitosis, which is the splitting and formation of 2 nuclei, we have cytokinesis, where the cytoplasm splits. We then start the cell cycle from scratch with the gap phase 1, where we have growth. I think of it as growth phase, but it's often called gap phase. But you've got growth of organelles, etc., blah, 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 blah. Then we have the synthesis phase, where the DNA doubles. And then we have, I've made this section a little bit too big. If anything, G1 should be big, but this is just a crusty little diagram, so don't worry. G1 is normally the biggest stage where we get the biggest increase in the size of the cell. Okay, G2, you've got the second DNA check, the gap 2. So that's the order. So you just got to find which one follows the order. So here we've got, we look at A, cytokinesis is 4. We should have G1 being 5. No, that's completely wrong. So this is wrong. Okay, let's look at B. They've got cytokinesis as 5. That would then mean G1 is 1, yes. S phase is 2. Now they've got the S phase as 4 after cytokinesis is 5. So this is not correct either. Let's look at C. If cytokinesis is 3, then G1 is 4. Then what have they got? The S phase is 5. This doesn't look right. Okay, which one of these is right? That don't look right either. What the hell? Let's look at D. If cytokinesis is 4, G1 is 2. No, that don't work either. All right, so far I've found that none of them work, so I've made a little mistake here, that one of these has got to be correct. It's got to go in this order. With my eyesight, it could be any of them. All right, let's see. G1, that's 1. Mitosis 2. No, that can't be right. Synthesis 4. Synthesis is between G1, so it should go G1, S, and G2. So let's check these. 2. No. 1. No. G1 is 4. Oh, it's this one. It's C. G1 is 4. S is 5. That makes G2 1. Mitosis 2. Ah, yeah, it's this one. So the order they're going here is 3 for that. Then G1 is 4. Then the S phase is 5. So we're going in the correct order. And then we've got G2 must be 1. And then mitosis is 2. So you can see 1, 2, 3, 4, 5. So it's in the correct order. It's just all over the place. Got myself in a muddle there. Boom. I hope you managed to follow that. If not, don't worry. But it all relies on you being able to get the order right down here. Super duper duper. You should learn that off by heart. If you don't know it, what you're doing, learn those basics off by heart. Let's move on. Keep moving on. All right, question 6. Hox genes contribute to the overall body plan of an animal. Which of the following rows correctly describes Hox genes? So Hox genes are clusters of genes that are vital for the body plan in animals, and Hox genes are similar between different species of animals. They're very, very similar. You only get small differences, 1 or 2% differences in the Hox genes as you go between species. So we can look at the Hox genes as they're relatively stable between all animals. We can look at those to study evolutionary relationships. However, this is not what this is talking about. So the base sequence varies between taxonomic. No, it's pretty similar between animals. So I wouldn't say that. What do the Hox genes transcribe? They code for a protein, normally a repressor or activator protein that is going to change. So yeah, similar in all animals. So we're looking at C and D because Hox genes are found in all animals and they're quite similar, generally 180 base pairs. The product is a polypeptide. That's true. It's like a repressor protein like we saw in the lac operon or in the operon. Have mutations have little or no effect. No, if you have a mutation in a Hox gene, that's where you could end up with your eyes on your bum cheeks or your ears on your feet. They're often lethal because they change the body plan. So you could end up with your arms where your legs are supposed to be and your legs where your arms are supposed to be. Most of these are often lethal. Not always, but often. So let's go for D here. That is the correct answer. Don't get your Hox genes mixed up with your homeo, whatever they're called, genes, which are found in plants, animals, and fungi. Okay, homeobox genes. Right. They're also to do with the body plan, but specifically they are in animals, plants, and fungi. Most eukaryotic cells, except for protists or protoctists, which are eukaryotic but single-celled, so don't need a body plan. So however, fungi, you think of a mushroom, it needs to know where its hyphae and stuff are and where the cap of the mushroom is. So fungi do require a body plan as well. So the three kingdoms that require body plans are plants, animals, and fungi. You're not going to need a body plan in protoctists or bacteria. Okay, Hox genes contain a homeobox sequence of 180 base pairs. Two species have a homeobox sequence of 180 base pairs, where 1.7% of the base pairs are different. Which of the following shows the number of amino acids coded for that would be different in the two species? So what I would say straight away is if we have 1.7% difference in the bases, we want to work out how many bases different that is. So 180, we want to find out, I'm going to times by 0.017, and that's just my little cheat code. So 0.017 times 180. So that gives me 3.06 base pairs are going to be different. So if there are 3 bases different between the species, how many different amino acids can you make from 3 altered bases? Well, that means they could all be in one codon, or the 3 different bases could be in 3 different codons, right? So you could potentially, if they're not silent mutations, you could potentially maximum, you could change 3 amino acids. So we're looking at a maximum of 3 amino acids. But what's the minimum? Okay, so when we're looking at the minimum, So I can get rid of this one here because we're changing 3 bases. So I must be able to have a maximum with 3 different bases out of the 180. If they're spread out, it's going to affect 3 different codons. Therefore, it could potentially code for 3 different amino acids. We're looking at substitution here, aren't we? If we look at B and D, which one's correct? Well, we know that DNA is degenerate. So hopefully you know what that means. DNA is degenerate. So DNA is degenerate. This means each amino acid has more than one codon or triplet code. So what that means is that although I might alter a base from, say, GGC to GCC, it doesn't necessarily code for a new amino acid. So actually, although the 3 bases have altered, there might be absolutely no mutations and no different amino acids. So the answer is going to be B, because the minimum will be 0. Because if I'm just swapping the bases, it's not a frame shift. It's not a definite change in the base sequence. And it's definitely not, it doesn't have to lead to different amino acids. So the answer is B. There's a minimum of 0 and a maximum of 3 different codons will be changed. If we change 3 bases somewhere along that DNA, then that could change up to 3 amino acids, a maximum. Okay, awesome. Hopefully you understood what I was waffling on about. Okie dokie, we're up to another question. Genes are not expressed during cell division because chromosomes are more tightly wound around the histone proteins than during interphase. Which of the following shows the level at which gene expression is being controlled when DNA is more tightly wound during cell division? So what you're looking at here is you have to understand what histones are. So your DNA is wound around histones. What that means is your histones, I think of them as like cotton wheels. They're actually proteins, and the DNA is wound around them. So if the DNA is tightly wound around them, oh God, I can't speak, then the RNA polymerase cannot access the DNA and transcribe the genes. So what they're saying is that the chromosomes are more tightly around the histone during cell division. This means that you can't transcribe the DNA. What is it that RNA polymerase can't do if it cannot access the DNA? If it can't get in and read the DNA, what can't it do? Well, it can't do transcription. So this is cellular control on the transcriptional level. Okay, by tightly winding the DNA around the histones, the bases are not exposed for the active site of the RNA polymerase to lock on and then start reading those bases. So this is transcriptional interference we got going on here. Translational would be when it's attached to the ribosome. Okay, and post-transcriptional would be after transcription. We're talking about interfering with splicing or the mRNA at that point. Okay, cool. Let's move on. Oh my good gosh, change. Change, I tell you. Why does this thing not flip on? 9, right. Question 9. Which of the following processes is important in determining the overall body plan of an organism? Now, we already talked about apoptosis, cell death being vital. So let's see if that, no cell death here. Endocytosis, so that's bringing things into the cell through the cytoplasm. Exocytosis, that is things leaving the cell, you know, when the vesicles, their membrane becomes one with the membrane on your cell and secreting things outside of the cell. Meiosis, that's the formation of gametes. It's really only involved in the formation of gametes. And mitosis is the answer. We know that apoptosis and mitosis are vital processes in the body plan development. Right. Here we go. We've got a couple of written questions to finish up. A gene mutation is a change in the sequence of nucleotides within a gene. So you've got a mutation in a gene. It's a change in the nucleotides within a gene. Explain how it's possible for a mutation to have no effect on the protein produced from that gene. Well, what did we just talk about earlier? If one of your bases has changed, does that definitely mean you get a new amino acid? It doesn't. Why is that? Okay. It could be a silent mutation. Okay, so you're going to get 1 mark for saying silent mutation. DNA is degenerate. So if you can remember that, you're going to get the second mark. Because DNA is degenerate, we can get a silent mutation. And what do we mean by degenerate? Well, that means that um, um, each amino acid has more than one triplet code. So a different base or nucleotide sequence could code for the same amino acid. So you won't get a change in the tertiary structure of or in the primary and then tertiary structure of the protein if you have the exact same sequence of amino acids. So, but you could just put silent mutation and DNA is degenerate to explain that whole bit underneath. All right, let's read the next bit. Explain how a mutation could alter the protein. So explain how a mutation could alter the protein so that it no longer performs its correct function in the cell. So how could a mutation alter that? Well, like this. A different base sequence or change in the gene, different change in a base could change the amino acid transcribed, so change the primary structure of the protein. Now, if you have a different primary structure, it means your R groups are in different positions and we get a different interaction, hydrophilic, hydrophobic, ionic, covalent, whatever. We get different interaction between those different R groups. And therefore, this alters the tertiary, oh my gosh, the tertiary or 3D structure of the protein. Now, if this is a hormone, maybe it can't bind to the target cell. This alters the 3D structure of the protein. The enzyme may no longer bind to the active site. And what am I on about? To the substrate. Yeah, I suppose I'm just putting an example. The enzyme's active site may no longer be complementary to the substrate. Okay, you might not form an enzyme substrate complex, so that protein will now be dysfunctional. Okay, so you can get dysfunctional proteins could result from any change in their 3D structure. So, for instance, hormones may no longer be able to bind to the receptors on target cells, like insulin can't bind to the insulin receptors on liver cells, things like that. So there we go. Yeah. Right. Let's move on. Okay, 16. Gene transcription. I don't know why it's got 16 there, but that's just the way it is. That's just the way it is. Gene transcription is controlled by transcription factors. Figure 16.1 shows how a transcription factor can control transcription. So we've got a hormone coming in here. We've got plasma membrane. Hormone is entering. We've got an inactive transcription factor just floating about. When that hormone randomly combines with that, obviously, if you have an increase in the hormone, you're going to get an increased probability of binding with this inactive transcription factor inside the nucleus. This activates the transcription factor, which seems to have joined to region A, which is allowing RNA polymerase to attach. And then we have, obviously, this is some sort of structural gene, which can now be read. So name site A. So think of the operon. We've got P, O, and we've got the structural genes. What do you think site A is? Well, it's where the RNA polymerase attaches. So this must be the promoter region. Now, you could have put the operator region because normally repressor proteins bind to the operator region. So you'd be forgiven if you made that mistake. However, this is an activating protein. This transcription factor is obviously an activator. And by binding here to this region, it has meant that the promoter region has become more complementary to the RNA polymerase's active site, allowing transcription to take place. So it's another way of controlling transcription here. Okay, using the information in figure 16.1, describe how transcription can be controlled in eukaryotes. So we've literally just got to describe what is happening here. So we can say, just describe what's in the diagram. Hormone binds to transcription transcription factor, which activates and binds to the O region, making the promoter region available for RNA polymerase, which can now transcribe the genes. So there we go. Really just describing what's happening in the diagram. Okay, what's this? 3 marks. Describe how gene expression can be regulated after transcription. Okay, so after transcription, so what happens in transcription? What do we create? Pause the video, have a go. All right, I'm going to go through the answer. So after transcription, we're going to make mRNA. So mRNA, and what happens to the mRNA? mRNA is spliced, removing non-coding DNA. What are those called? Introns. So essentially, you're going to get 2 marks here for saying that the mRNA gets spliced, which means non-coding parts of the DNA, the introns are removed. It can then bind to the ribosome, and be translated into a polypeptide, because you're only left with the structural genes, which can now be read and translated into the polypeptide once you have removed the introns. Super. Let's move on. Rapidly moving on, right? We're nearly finished here. So we now have a fill in the gaps. The body plan of multicellular organisms is under genetic control. Complete the passage below using the most appropriate word from the list. So, see these words here. Try to see if you can work out which space the words go into. So the development of the body plan in eukaryotic organisms is controlled by something genes. Okay, try to fit the appropriate word. Pause the video, have a go. All right, I'm going to go through the answers. So the development of the body plan in eukaryotic organisms is controlled by the homeobox genes. Now, you may have wanted to say Hox genes, but I don't think it was there. They'd have made it a little bit more tricky if they put Hox genes there. But of course, Hox genes, the clusters of Hox genes are only found in animals, and this was specific, saying eukaryotic organisms, and eukaryotic organisms have homeobox genes. These genes code for proteins that are able to bind to something and turn specific genes on and are known as something factors. So what is it that the proteins from the homeobox genes, what do they code for? What type of proteins? And what do those proteins bind to? So you remember the repressor protein. Where did the repressor protein bind? What did it bind to? So the repressor protein binds to the operon. Let's see, does it have operator region? Let's just go operon. And turn specific genes on and off, and are known as something factors. What we're looking at here is transcription factors. Now, transcription factors can either increase or decrease the rate of transcription, depending on what they do. These proteins contain a sequence of base pairs that varies little between species within the animal, something, or fungus. What were the 3 eukaryotic kingdoms, guys? So we've got animal, and we're looking at plant or fungus kingdoms. And there are your answers. So homeobox, operon, transcription, plant, and kingdoms. Super. Hope you got 5 out of 5.
You're still here? It's over.